Welcome back, students, to the food hazards, and today we're going to talk about the microbiological hazards. The microbes presence in food as well as in water is quite familiar to us, and we do check for the analysis of the presence of pathogens, usually in the case of food as well as in water. And these pathogens could cause mainly two different types of illness. It could be either a foodborne infection or it could be a foodborne intoxication. Now, the ingestion of microbes followed by their growth and tissue invasion as well as their release of toxins sometimes in the body would result in the formation of foodborne infection. So, in a foodborne infection, you always need the presence of the microbe within your body. And the subsequent events could result in the formation of a foodborne infection. But sometimes it could be another case, sometimes like called the foodborne intoxication. In such cases, the ingestion of the toxins in the food in which the microbe has grown, that is, previously the microbes have grown and they have already been produced some toxins in the food. So the taking of that particular food which has already been intoxicated or which is already having a toxin, a microbial toxin, that could result in the formation of a foodborne intoxication. They mainly include like instances you have Staphylococcus food poisoning, botulism, Clostridium perfringens food poisoning, Bacillus cereus food poisoning. These are some examples that which we have. So the foodstuffs should not contain the microorganisms or the toxins or metabolites in quantities that present an unacceptable risk for human health. That is a main priority that we should keep in our mind when we do a microbiological analysis. Now, how do we do the microbiological analysis? You take a food, you homogenize it, you solubilize it. If it is a insoluble form, you make it into a soluble form, you separate it, and you take the food analysis and you do the quality control. Now, if you go to consider the different types of food types which we have, and it has been found that different foods will cause a different range of food illness. And coming to the protein, if you go to have a protein rich foods, they are the ones where the microbial growth has been found to be more than like poultry, dairy and beef. They are being found to be a good source of food illness. That is a predominant source of food illness. So you might think, why egg is not here? Egg is a different factor which has a shell which protects it from microbial infestation. And of course, the leafy greens and the fruits and the nuts could also be a cause of food poisoning, mainly because of the fecal contamination which could be possible in the leafy greens. The microbiological hazards, as we mentioned earlier, they could be of either the microbes or they could be by the microbial toxins, and the microbes could be either of bacteria, fungi as well as viruses. Coming to the bacterial hazards, we have different types of bacterial hazards. They could include Escherichia coli, the common fecal contaminant, Salmonella typhi, which causes typhoid, Campylobacter jejuni, Listeria monocytogenes, Bacillus cereus, Clostridium perfringens, and Shigella flexneri, Vibrio cholera, the O1 and sometimes non-O1 also, and Vibrio parahemolyticus, Streptococcus pyogenes, Yersinia enterolytica, Yersinia pseudotuberculosis, Clostridium botulinum, and this list is just in fact insufficient because there are lots of pathogens, bacterial hazards, which could be present in different types of foods. Now, the CDC, the Center for Disease Control, reports as per the US reports in 2000 level, it was being found that norovirus constituted for about 58% of the foodborne illness cases in 2011, and salmonella. So among the viruses, the predominant cause was norovirus, and among the bacteria, salmonella was a predominant cause of foodborne illness with 11% of reports in 2011. And the causative of death via food, The first place goes to Salmonella with 28%. The second place goes to Toxoplasma gondii. The third place goes to Listeria monocytogenes, as well as the fourth place goes to norovirus. So you should remember, of course, it could cause illness. Norovirus causes more illness, but the most death is being caused by Salmonella. Something which is of much importance when you consider a food industry is this growth curve of a bacteria. Whereas we know that there is always a lag phase, an exponential phase, a stationary phase, and a death phase for the bacteria. So you have to maintain, if you need to control the microbial load, all the processes, all the food preservation processes, it should be in such a way that either you delay this lag phase or you never let it enter into the exponential phase. So all the preservation strategies or all the control strategies should be in such a way that the extent of the lag phase should be increased, or even the killing of the microbe shall be done. Now, if you cannot even kill it, the basic strategy that you should have, at least you should increase the lag phase of the bacteria. If a bacteria is entering into the exponential phase in a food, it indicates that could lead into the rapid multiplication of the microbe within the food, and the microbial load of the food would increase. So every strategy that we have to take is to increase the lag phase of the microbial growth in the food. Now, I've just given you some examples of what are the factors which affect the growth of some food pathogens. Now, three main factors which affect the growth of foods. We have already mentioned this in our previous sessions, but I just want to remind you, of course, the temperature is something important. At the temperatures beyond the optimal levels, beyond either above or below, the growth of the microbes are being inhibited. And pH is something, so you can prevent the microbial spoilage by controlling the pH. Okay, and the water activity. The water activity is something which would refers towards the availability of water for the microbial growth. By controlling these three factors, we can in fact control the microbial growth. We have already mentioned this in the respective sessions, so I am not just going into the deeper of this. And most of the bacteria, they prefer foods that are slightly acidic or have a neutral pH. So ranging from about 4.6 to 7.5, and many require oxygen. And they also need moisture to grow, and they usually thrive at temperatures of about 5 degrees centigrade to 57 degrees centigrade. And any temperature inside this region is considered to be as a danger zone. So you have a temperature range like this, and if a microbial, if you keep the food at a temperature between these two, at these conditions, probably the microorganisms grow at a very rapid rate. And considering the food quality, the temperature of 5 degrees centigrade to 57 degrees centigrade is considered to be as a danger zone. So we should always try to prevent the occurrence of food at temperatures beyond this, like between this. Like if it is a raw food, okay, I'm talking about a raw food. A raw food should never be kept at temperatures between this, because it would result in the microbial multiplication within the food. Then, of course, if you are keeping it with various other preservatives, that is a different case. Okay, and the pH range is also something important. If you lower the pH of the food, as we have mentioned earlier, the chances of microbial growth will be found to be less. And as per the 2014 Food Safety Progress Report, you can see that there is a 13% increase in Campylobacter based infestations, and E. coli O157 there is a 32% decrease, whereas in Vibrio as well as in Yersinia you can see there is a different range. Vibrio infestations have been found to be increased, and 22% Yersinia has been found to be decreased. But something of very much importance is Salmonella. There is no much change, and we had mentioned earlier that you can see the death is mainly caused by Salmonella species, and it occupies the first place in the extent of death which has been caused by Salmonella. So much care has to be taken to prevent the infestation by Salmonella microbe. So these are some different microbes. You can, in fact, almost most of your organisms would share similar foods, and you can write them in different ways. Like sometimes you can also see Salmonella in fresh produce also, as well as you can see them in also in bakery foods. Now you can see sometimes we have the Salmonella infestation in the shawarma. Okay, so that's all different. You cannot give a strict rule, right? So much microbes, these microbes could be found in this particular food because they share different habitats, and a change could be occurrent in that. So now we'll be going into the details of each and every microbe in a very brief manner in this coming sessions. Hope so you got an idea on the microbial hazards. So I'm coming to an end with this slide. Thank you for now.